Hello everyone, today's presentation is on transversely isotropic biological models for tissue. Our research was driven by the desire to explore models for anisotropic materials. Specifically, we focus on understanding how biological tissues, like skin, can be represented using mathematical models. One ideal case is transversely isotropic materials. These materials exhibit symmetry about an axis that is perpendicular to the plane of isotropy. We conducted biaxial tension tests on samples of chicken and turkey skin using a planar biaxial testing system. Each test involved varying displacement ratios in the x and y directions, resulting in a maximum principal stretches at angles ranging from 0 degrees to 90. From the collected force and displacement data, we calculated the PLO stress. Our approach involved utilizing invariant-based models derived from constitutive models proposed in previous research papers. These models account for both the isotropic effects of the matrix and the anisotropic effects of the fibers.
These are the architecture of two models we're comparing. The left one is the previous model introduced in the lecture. Four invariants are connected to linear and quadratic terms and then to identity and exponential. This model also predicts alpha the fiber direction. So with 16 functions, we have 60k possible models. Our model is on the right side. We've replaced the exponential of squared invariant with ln of that which appears on null models. We also introduced I1 times I4 terms. So with 23 functions, we have 8 millions of possible models. Notice that we used PLO stress while the previous model uses Cauchy stress because we obtain stress by dividing force with undeformed area resulting in engineering stress. These are the results using pigskin data. With the previous model, we got R2 scores shown here with all the I1 terms activated. Then with our model, we got higher R2 scores than the previous model. The activated terms are listed here, showing the mixed term explains mechanics not included previously.
We now move on to the avian portion of our results. With chicken skin data, the previous model yields the following R2 scores with 3 activated terms. With our new model, there are now 4 different activated terms, and although the R2 score in the x direction is now lower, the R2 score in the y direction increases. Turkey skin data with the previous model shows the same terms as the chicken skin, with the R2 scores shown here. The new model, however, has many more activated terms, again explaining mechanics not covered by the previous model. And although the R2 score in the x direction is lower, the y direction increases significantly from 0.656 to 0.845. In conclusion, we have demonstrated how constitutive artificial neural networks can be used to understand the mechanical properties of avian and porcine skin. Future directions include expanding the study to other species, such as bovine or ovine, and also to add more published constitutive models to our network to better predict skin's mechanical properties.